features of Oracle. At Oracle, as you all know, we have live classes, recorded classes, notes, and MCQs, all based on the latest syllabus. Apart from that, we have personalized counseling sessions. So you can anytime book a slot with us for any extra guidance and support that you need from our end. We have 3 different subscription plans at Oracle, beginning from basic, which is for 15,000,
classes, notes, and MCQs, all based
and it is for 3 months. Then we have standard plan, which is for 30,000, and it is available for 6 months. Then we have mentorship plan, which is a premium subscription, which is for 50,000, and it is available for 12 months. So it is a year-round subscription. In this, we have live doubt clarification sessions and also one-to-one personalized guidance. So what is this internal exam marathon series? In this, there will be rapid revision of all important topics. Okay, so basically anything that can be asked in the exam will be dealt in this series. Apart from that, there will be volatile quick revision, facts revision, then multiple choice questions will also be covered. So we assure you that you get top-notch grades in your exams. Okay, so for today's class, I've decided to take these cycles from carbohydrate unit. So the most important cycles that you have in carbohydrate metabolism, they are your glycolysis, gluconeogenesis, glycogenesis, glycogenolysis, TCA, and HMP. So these are the very important cycles of your carbohydrate metabolism. And there is equal probability of any of these cycles being asked. So I cannot tell you you can just study glycolysis and skip gluconeogenesis. No, there is equal probability of any of these cycles being asked. Then you have Rappaport and Corey cycles. They are also important and quite frequently they are asked in short notes for 5 marks. You might get them. Now your fructose, galactose, and lactose metabolism, they are less asked. Okay, but you should be knowing there are clinical applications to this fructose intolerance, galactose intolerance. There's nothing when I say important and unimportant, so by unimportant, I don't mean that you should not be knowing those things. All I mean is in relative terms, okay? Compared to the other cycles, they are not as frequently asked as your glycolysis and gluconeogenesis and all. So we'll be taking each of these cycles, and I've decided to take these cycles under the following heads. So these are the main headings that will be covered, and they're like the bare minimum you should know for all the cycles. That is, you should know the correct pathway of the cycle, that is the substrate and the enzyme and the product. The correct sequence you should know at least for all the cycles. That is like the minimum threshold that you should be knowing. Then the site or the organelle involved. Apart from that, whether it is taking the fed or the fasting state. Then the energetics is important. Sometimes you only get asked about the energetics. Or regulation is really very important. Okay, so regulation is one thing you cannot skip. And then of course clinical, we studying everything for the sole purpose of studying its clinical. Okay, so and in miscellaneous, I've decided to take some extra relevant information if there is for that particular cycle, that will be covered in miscellaneous. So this was a concept map of the class, that how the class will be conducted. So for today's class, I'll try to cover the maximum number of cycles. Let's see how many of these cycles we're able to cover in this class. Okay, so without wasting much time, let's begin our class. So glycolysis, we will begin with glycolysis. So covering the first heading, that is the cycle. So this chart, this flow chart for all the pathways, this is like the minimum you should be knowing. That is, you should know the substrate, you should know the product, and the relevant enzyme for that step. Okay, it is not necessary for you to know the steps and details. You'll not be asked to describe. If you draw this chart, if you get the pathway correct with the correct steps showing the ATP involved, if there's any ATP involved or generated or used, or any reducing equivalent being generated or produced, along with that, if you get the products and the substrate and the enzyme correct, it will fetch you marks. Okay, so let's begin. So for glucose, we had studied that after you've had your meal in the well-fed state, what the blood glucose level increases. Now this glucose enters the cell. And if you remember in the regular classes, we had seen that glucose was entering the cell through glucose transporters. Okay. Those glucose transporters, they were bidirectional. So the glucose that was entering the cell can also move out of the cell. Therefore, this first step was the trapping. The significance of this first step was trapping of glucose. Now this glucose, it is phosphorylated by hexokinase, and another enzyme was glucokinase. It is phosphorylated to form glucose 6-phosphate. And for this step, one ATP is required. Where is this phosphate coming for phosphorylation? It's coming from the ATP. So glucose changes into glucose 6-phosphate. The enzyme involved is hexokinase, and one ATP is required. So this is like the minimum you should know for this step. Followed by now this glucose 6-phosphate, it isomerizes to form fructose 6-phosphate. So you know that isomerization is taking place. So the enzyme will be isomerase. Next, you know that another phosphate is being added to this fructose 6-phosphate. So you need a kinase enzyme, okay? And this phosphate is also being provided by, again, ATP. So one ATP is used here. Now this fructose 1,6-bisphosphate, it is split by aldolase. Now to form glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now this dihydroxyacetone phosphate, it again isomerizes to form glyceraldehyde 3-phosphate. So you have two molecules of glyceraldehyde 3-phosphate. And the next step from here onwards, every step will be into two. It will be happening twice because we have two molecules of glyceraldehyde 3-phosphate. And why is your dihydroxyacetone phosphate changing into glyceraldehyde 3-phosphate? Because the next enzyme, that is glyceraldehyde 3-phosphate dehydrogenase, it can only act on glyceraldehyde 3-phosphate. Now what happens, this glyceraldehyde 3-phosphate, it is acted upon by the enzyme glyceraldehyde 3-phosphate dehydrogenase to form 1,3-bisphosphoglycerate. Okay. And in this step, you know that dehydrogenases, they cause oxidation reduction reactions. Okay. So it removes a hydrogen from the substrate, that is glyceraldehyde 3-phosphate, and gives it to what? NAD plus. So NAD plus changes into NADH, and what you get as the product? You get 1,3-bisphosphoglycerate. Now this 1,3-bisphosphoglycerate is acted upon by 1,3-bisphosphoglycerate kinase to form what? 3-phosphoglycerate. And in this step, one ATP is formed. So this was the first substrate level phosphorylation. Okay. Now this 3-phosphoglycerate, it will change into 2-phosphoglycerate. So from the position 3, the phosphate is going to position 2. So this is an isomerization reaction, and this is taking place within the molecule. So the enzyme is mutase, phosphoglyceromutase. Now this 2-phosphoglycerate, it changes into phosphoenol pyruvate. This was a dehydration reaction. A double bond is being introduced, okay? In phosphoenol pyruvate, there's a double bond. This phosphoenol pyruvate then is acted upon by pyruvate kinase to form your pyruvate. So this is how your glucose is converting into pyruvate via glycolysis. So these, as I said, substrate, product, and the enzyme for each of these steps, okay? The minimum that you should know. Now, the glycolysis was of two types, aerobic glycolysis or anaerobic glycolysis. So, aerobic glycolysis is when oxygen was present, okay? So, it forms acetyl-CoA. And anaerobic glycolysis, when oxygen is absent. So, it forms lactate. And this is taking place in the RBCs, okay? And also, this step is important in the RBCs because of the regeneration of NAD. Because in the RBCs, there is no mitochondria. So there is no regeneration of NAD plus through the electron transport chain. So this step becomes really very important in the RBCs because it regenerates NAD+. So that was briefly about glycolysis, the pathway. Now moving on to the site, the glycolysis is taking place in all the organs and it is a cytoplasmic cycle and it takes place in the well-fed state under the influence of the hormone insulin. So after you've had your meal, your blood glucose level increases and that is when it needs to be brought down. So what happens, your insulin is released and it brings about the functioning of glycolysis. Glycolysis takes place. Now energetics. So you have two different energetics. One is for aerobic glycolysis and the other is for anaerobic glycolysis. In aerobic glycolysis, you have a net of 7 ATPs being generated. How? You have 4 ATPs being generated in 2 substrate level phosphorylation. This was the first substrate level phosphorylation as you can see here. And this step gave you the second substrate level phosphorylation. Okay. Now, as I said, that everything is happening twice because there are 2 glyceraldehyde, 3 phosphate molecules. So you get 4 ATPs. Okay, 4 ATPs are being generated. And how many NADH are generated? Here in this step, it gives you 2 NADH from 2 glyceraldehyde 3 phosphate molecules. So when this NADH, it enters your electron transport chain, it gives 5 ATP. So you are getting a total of 9 ATPs. Okay. Now out of 9 ATPs, you have to subtract 2 ATPs because 2 ATPs have been consumed. In which step? In the first and the third step. See, in the first step, hexokinase is using 1 ATP for phosphorylation, and then your phosphofructokinase is using another ATP for phosphorylation. So, 2 ATPs have been consumed. So, you subtract that and you get a total of 7 ATPs. Okay. So, in glycolysis, aerobic glycolysis, 7 ATPs are being generated. And in anaerobic glycolysis, you don't have electron transport chain running. Okay. Because there is no oxygen. In that case, what happens? Your lactate is being formed. So what do you have? 4 ATPs being generated and 2 were being consumed. So a total of 4 ATPs being generated. Okay, you don't get the ATPs that were those 5 ATPs coming from NADPH that are not available in case of anaerobic glycolysis. Now moving on to the regulation. Regulation of any pathway, any cycle is very important. Okay, the regulatory enzymes here are in glycolysis, they are hexokinase, phosphofructokinase, and your pyruvate kinase. These are the key regulatory enzymes of glycolysis. And as I said, that glycolysis operates under the influence of insulin. So what does insulin do? It dephosphorylates enzymes. So these regulatory enzymes, they will be active in dephosphorylated state. They are active in dephosphorylated state. Okay. Your hexokinase, it is inhibited by glucose 6 phosphate. Your glucokinase, it is activated by insulin and it is inhibited by glucagon. Your pyruvate kinase, it is activated by insulin and fructose 1,6 bisphosphate and it is inhibited by glucagon, ATP and cyclic AMP. And phosphofructose kinase, it is activated by insulin, AMP, fructose 6-phosphate, fructose 2,6 bisphosphate. And it is inhibited by glucagon, ATP, citrate, low pH, and cyclic AMP. So basically, your low energy states, that is AMP, it is activating your phosphofructose kinase, and your high energy, when there's already more energy, ATP, so your glycolysis will be inhibited. So ATP will be inhibiting your glycolysis. So that was about the regulation. Now moving on to the inhibition. Iodoacetate, fluoride, and arsenate. So iodoacetate, it inhibits glyceraldehyde 3-phosphate dehydrogenase. Fluoride, it inhibits enolase. This is important. You get a clinical question from this. And your arsenate, it inhibits 1,3-bisphosphoglycerate kinase. Okay. Now the clinical, that is the disorders associated with glycolysis, they are very rare because glycolysis is a very important pathway. So if your glycolysis is defective, it is very difficult for the child to survive. So any not many disorders are associated with it. Now, sometimes you get in reasoning questions, you might get asked why, when collecting blood samples, fluoride is added to blood samples. Explain why. Why? Now you know the answer, because when you add fluoride to blood samples, what gets inhibited? Enolase. So your fluoride is an inhibitor of enolase. When fluoride inhibits enolase, what happens? See, this step is inhibited. Now what will happen? Your glucose will not be metabolized to pyruvate, okay? And thus it will prevent giving lower readings of glucose. In case your chloride was not added, then you would have gotten lower values of blood glucose. Therefore, while collecting blood samples, you add fluoride to the samples in order to inhibit the enzyme enolase, and this you will not get lower blood glucose readings. So that was very briefly and quickly about glycolysis. In the similar manner, we will be covering the rest of the cycles. So you need to know the cycle, the pathway, then the site, organelle, energetics, regulation, inhibition if any, and the clinical associated with it. So let's quickly move on to gluconeogenesis. If you have any queries, please you can ask. So we've covered glycolysis. Okay, now we're going to talk about gluconeogenesis. And again, we'll be taking gluconeogenesis also under the same headings as glycolysis. So in gluconeogenesis, what is happening? You are producing glucose from non-carbohydrate sources. So gluconeogenesis is the production of glucose from non-carbohydrate sources. Now, all the irreversible steps of glycolysis, they are circumvented in gluconeogenesis via different enzymes, okay? And those enzymes which bypass or circumvent the steps of glycolysis, they form the key regulatory enzymes of gluconeogenesis, okay? Now, let's have a look. Your pyruvate changing into oxaloacetate. This is brought about by pyruvate carboxylase. What was happening in glycolysis? Your phosphoenol pyruvate here was changing into pyruvate. Now what do you have to do? You have to do the reversal in gluconeogenesis. But this enzyme here, pyruvate kinase, this enzyme of glycolysis, pyruvate kinase, it is an irreversible enzyme. So you have to bypass, circumvent the steps. This is happening in gluconeogenesis. How? First, this pyruvate, it changes into oxaloacetate, and then this oxaloacetate changes into phosphoenol pyruvate. This is how this step is bypassed. So for changing your pyruvate into oxaloacetate, you need the enzyme pyruvate carboxylase. This enzyme is needed. And for oxaloacetate to change into phosphoenol pyruvate, you need phosphoenol pyruvate carboxy kinase here. Okay, so what you will get now? We get phosphoenol pyruvate. Now from phosphoenol pyruvate, you get 2-phosphoglycerate using the same enzyme as it was used in glycolysis, okay, because that was a reversible enzyme. So all the reversible steps, then from 2-phosphoglycerate, it forms 3-phosphoglycerate, then 1,3-BPG is formed, then glyceraldehyde 3-phosphate is formed. So all these steps they were reversible, and until you reach fructose 6,1,6 bisphosphate. Now fructose 1,6 bisphosphate to fructose 6-phosphate, this will have to be circumvented. For this, you need the enzyme fructose 1,6 bisphosphatase. Remember, in glycolysis, what was happening? When fructose 6-phosphate was changing into fructose 1,6 bisphosphate, which enzyme was used? Phosphofructose kinase was used. Okay. This phosphofructose kinase was used. Now what happens? This phosphofructose kinase, this was an irreversible enzyme. So in gluconeogenesis, you have to bypass this step. So this is bypassed by fructose 1,6-bis phosphatase. Okay. Now you get fructose 6-phosphate. Now this fructose 6-phosphate again by isomerase, it will change where hexokinase was involved. Now hexokinase again was an irreversible enzyme, so we need to bypass that by using glucose 6-phosphatase. So glucose 6-phosphatase will be used and glucose is formed. So we have seen how gluconeogenesis operates by circumventing the irreversible steps of glycolysis. So this is the minimum you should know about gluconeogenesis. If you get asked about gluconeogenesis, you will have to draw these steps very carefully, okay? Showing the irreversible enzymes and then the enzymes of gluconeogenesis which bypass the irreversible enzymes of glycolysis. So this is again a repetition that how your pyruvate was changed into oxaloacetate. This was brought about by pyruvate carboxylase, and this was a carboxylation reaction, biotin-dependent carboxylation, and 1 ATP was used. Biotin was also involved, and carbon dioxide was incorporated. So a 3-carbon pyruvate was changed into a 4-carbon oxaloacetate. Now this oxaloacetate, it was acted upon by phosphoenol pyruvate carboxykinase. A carbon dioxide molecule was liberated. For this step, GTP was involved, and you get a phosphoenol pyruvate. And the pyruvate kinase step of glycolysis was circumvented until you reach fructose 1,6-bisphosphate. This was acted upon by fructose 1,6-bisphosphatase, and then where you get fructose 6-phosphate. Then again, isomerase was used. That was common in glycolysis as well. And you get glucose 6-phosphate. This glucose 6-phosphate was acted upon by glucose 6-phosphatase. And then ultimately your glucose is formed. Now the site where gluconeogenesis is taking place, it takes place in the liver and the kidney. And the organelle, it is both cytoplasmic and mitochondrial. Now your gluconeogenesis will be taking place in the fasting state under the effect of the hormone glucagon. Okay. Obviously, you do not need to generate glucose when you already have glucose. Okay, it will only take place in the fasting state when the body does not have glucose. That is when you need glucose. So the body will synthesize glucose in the fasting state under the effect of glucagon. One thing always remember, how will you remember that which is the hormone of fasting and which is the hormone of fed state? Always remember glucose gone. When glucose is gone, that is the starving state, okay? Starving. Then you have glucagon. So this is the important thing. Just remember glucose gone, then you have the hormone glucagon. So automatically then you will remember that in the fed state, the hormone is insulin. So this is very important, okay? Don't ever get confused between these two enzymes, okay? Hormones, sorry. Now energetics. 6 ATPs are used, okay? So there is consumption of ATPs while you generate 1 mole of glucose. 6 ATPs are used how? See here. This step pyruvate carboxylase. This was using 1 ATP, and since your pyruvate is 3 carbon and your glucose is 6 carbon, so you will be needing 2 pyruvate molecules to generate 1 mole of glucose. So here you will use 2 pyruvates. So therefore this for this step your 2 ATPs used, okay? Now again oxaloacetate to phosphoenol pyruvate, again 2 ATPs used. So you have used 4 ATPs. Now this step here in glycolysis, when 1,3-BPG was changing into 3-phosphoglycerate, your substrate level phosphorylation was taking place and ATP was being generated. So 2 ATPs were being generated there. Now when you're doing the reversal, you will need to give 2 ATPs. So 2 ATPs used here. So this makes a total of 6 ATPs. So this is the energetics of gluconeogenesis, that 6 ATPs are being consumed to produce one glucose molecule. Now, regulation. Moving on to the regulation, you have three different types of regulation for gluconeogenesis. That is your covalent modification, allosteric regulation, and reciprocal regulation. Covalent modification, we all know it is hormonal. It is brought about by the hormones insulin and glucagon. So here in this case, your glucagon activates regulatory enzymes by phosphorylating them. As I just told you that your gluconeogenesis will be taking place in the fasting state. So in the fasting state, the hormone involved is glucagon. So your glucagon, it phosphorylates the regulatory enzymes to activate them. So your key regulatory enzymes, as we've discussed, they will be active in the phosphorylated state by glucagon. Now, allosteric regulation, it is brought about by acetyl-CoA. Your acetyl-CoA, it activates pyruvate carboxylase. So, acetyl-CoA is an activator of pyruvate carboxylase, but acetyl-CoA is never a substrate for gluconeogenesis. Always remember that. Now, reciprocal regulation. It is brought about by a bifunctional enzyme, phosphofructose kinase 2. We have seen about phosphofructose kinase 1 in glycolysis. Now, this phosphofructose kinase 2, it brings about regulation, reciprocal regulation of glucogenesis and glycolysis. What happens is, your phosphofructose kinase 2, under the effect of insulin, when it is dephosphorylated, so in the dephosphorylated state, this kinase part of this tandem enzyme is activated. So when this kinase part is activated, it adds phosphate to the position 2 of fructose 6 phosphate to form fructose 2,6 bisphosphate. And this fructose 2,6 bisphosphate is an important allosteric, it is an important activator of phosphofructose kinase 1. So what will happen? Your glycolysis will be activated under the effect of insulin. Now, when there is glucagon and this tandem enzyme is phosphorylated, so in the phosphorylated state, this tandem enzyme, this phosphatase part is active. So when the phosphatase part is active, your fructose 1,6 bisphosphate, it will be changed into fructose 6 phosphatase. Okay, so this fructose 2,6 bisphosphate will not be there to activate phosphofructose kinase 1. So your glycolysis will not be activated. And what will happen? Your gluconeogenesis will take place in that case. So that was about the regulation of gluconeogenesis. So these are the important points that you need to remember. Like if you get a question on regulation, so you should know that there are three different types of regulation: covalent modification, allosteric regulation, and reciprocal regulation. Now, what is happening in covalent modification? Which enzyme is involved? What are the regulatory enzymes, and in which state those regulatory enzymes are being activated? That is in the phosphorylated state or in the dephosphorylated state. Moving on to the allosteric regulation, acetyl-CoA being the most important allosteric activator of pyruvate carboxylase. Then you should know about reciprocal regulation, which is being brought about by bifunctional tandem enzyme phosphofructose kinase 1. Then you should know then which part of this tandem enzyme will be activated under the effect of insulin and which part will be deactivated under the effect of insulin, and which of it will be activated under the influence of glucagon, and what are its effect. Now, what is the significance of gluconeogenesis? It is to maintain the blood glucose level during starvation. Then, sometimes you might get asked about substrates of gluconeogenesis, that is your lactate. When exercising muscles, there's a lot of production of lactate. So in order to avoid accumulation of lactate and in order to avoid muscle fatigue, that lactate is being converted, is entering gluconeogenesis to being converted into your glucose. Then glucogenic amino acids, primarily alanine. Then you have glycerol, part of fatty acids, of fats. Okay, when your fats, they hydrolyze, what happens? Glycerol and fatty acids are being formed. So the glycerol of fats, that is one of the substrates of gluconeogenesis. Then propionyl CoA is also one of the substrates of gluconeogenesis. It enters at the level of succinyl CoA. So that was about gluconeogenesis. Now let's see what is the next cycle that we have. So we've covered glycolysis, we've covered gluconeogenesis. Now we will talk about glycogenesis, that is synthesis of glycogen, and glycogenolysis, that is breakdown of glycogen. So in glycogen synthesis, what are the main steps in glycogen synthesis? That is synthesis of UDP glucose followed by branching. Okay, so these are the main steps in glycogen synthesis. First is synthesis of UDP glucose. Why do you need to synthesize UDP glucose? Because UDP glucose is the source of the glucosal residues that are being added to the glycogen primer to form your glycogen. Firstly, there is one question associated with glycogen synthesis, that is why can you not store glucose as such and you need to convert it into glycogen? Because your glycogen is a highly branched polymer, so it becomes really compact. Okay, so it will occupy a much lesser space. Secondly, your glucose is able to absorb water and the cell will swell up. So these are the two reasons why you store glucose in glycogen and not as glucose as such. Now, what is glycogen? We've already discussed this, that glycogen is a highly branched polymer. The straight linkages are alpha 1,4 linkages, and at the branch point you have alpha 1,6 linkages. Now let's begin with the synthesis of glycogen. So the first step in glycogen synthesis is the synthesis of UDP glucose. What happens here is your glucose 1-phosphate, it combines with uridine triphosphate to form UDP glucose and a pyrophosphate is released. And this is brought about by the enzyme UDP glucose pyrophosphorylase. So the alpha D glucose attached to the uridine diphosphate is the source of all the glucosal residues that are added to the growing glycogen molecule. That is why it is important for you to synthesize this UDP glucose, because this UDP glucose will actually be the source of glucose that will be added to the glycogen chain. Now what happens once you have synthesized your UDP glucose? You need a glycogen primer. To this glycogen primer, you will add UDP glucose. This UDP glucose will provide glucose to this glycogen primer. And then by the action of glycogen synthase, your glycogen will be synthesized. And how is branching taking place? See, this is your glycogenin. What is glycogenin? This is the primer we're talking about. Why do we need a primer? Because your glycogen synthase cannot act glucose molecules to a free glucose as such. Okay, it needs a primer. So what is a primer? This glycogenin is a primer, or a small fragment of glycogen, already existing glycogen, can also act as a primer. To this, your glycogen synthase is able to act glucose residues. So you see, this is the primer. To this primer, your glycogen synthase from this UDP glucose is adding glucose residues. So this was the primer. This is the primer. To this primer, you can see the one in red. They are the glucose molecules that are being added from the UDP glucose. So by the action of glycogen synthase, these have been added. Now comes the action of branching enzyme. What will branching enzyme do? It will transfer 6 glucose residues from this end, 1, 2, 3, 4, 5, 6. Or 6 glucose residues are transferred. So you see, they are transferred here and it forms alpha 1,6 linkage. A branch point has been created here. Okay, now this will repeat several number of times until you get this highly branched glycogen structure. Okay, so this was about synthesis of glycogen. And where does it take place? It takes place in the liver and muscle. The organelle involved is cytoplasm, and your glycogen synthesis will be taking place in the well-fed state. Okay, when you have excess glucose, then you need to store that glucose. Okay, so it will take place in the well-fed state under the effect of the hormone insulin. That is when your glycogen synthesis is taking place. Now you might get asked about this primer, glycogenin. So what is glycogenin? It is a homodimer. That is, it has two identical monomers. And these two identical monomers, they will autoglycosylate each other until 7 to 8 glucose residues long, as you can see here in this picture. See, 7 to 8 glucose residues are already being added. Now, this acts as the primer. Now, on this primer, the glycogen synthase will act and it will add glucose residues from UDP glucose until 7 to 8 long glucose residues have been added. 11 to 12, okay? Now what happens? The branching enzyme will come and it will transfer almost a 6 residue long unit from this end to another point to form alpha 1,6 linkage, and a branch point has been created. So that is how your glycogen has been synthesized. Now moving on to glycogenolysis. When will your glycogenolysis take place? It will take place in the early fasting state when the body does not have glucose. So your glycogen will be broken down to provide glucose. Now what are the steps in glycogenolysis? The first step is action of glycogen phosphorylase, followed by removal of branches, then conversion of glucose 1-phosphate to glucose. Now, what is your glycogen phosphorylase doing? See this diagram. This is your glycogen. Okay, now your glycogen phosphorylase, it will sequentially remove. Okay, see this diagram I'm drawing here. This is your glycogen. So your glycogen phosphorylase will act and it will sequentially remove these glucose molecules as glucose 1-phosphate. Okay, so glucose 1-phosphate, glucose is not released as glucose by glycogen phosphorylase. It is removed as glucose 1-phosphate. Okay, see this. These are all glucose 1-phosphates being released. Now what happens? Until it is 4 units away from the branch point. Now what will happen? Transferase enzyme will come into play and it will transfer a trisaccharide unit. You can see this. It will transfer this trisaccharide unit here. See, this trisaccharide unit has been transferred here if you see this. Now this one glucose that is left at the branch point, it will be removed by the debranching enzyme, and this is removed as glucose. Then again, what will happen? Your glycogen phosphorylase will further act. It will sequentially remove glucose 1-phosphates unit. So this is how your glycogen is being broken down. So even if you don't write full paragraphs, and that is how you attempt the questions, okay? Flowcharts and diagrams are more than enough. If you get this correct, it is self-explanatory. You don't even need to explain if you get this right. Once again, let's have a look. Let's suppose this is your glycogen molecule. Okay. Your glycogen phosphorylase will act on this and it will sequentially remove glucose as glucose 1-phosphate until it is 4 units away from the branch point. This was the branch point, 4 units away from this branch point. Now what will happen? Debranching enzyme will come, which is a tandem enzyme. It is a bifunctional enzyme. It has two functions. One is glucan transferase and the other is glucosidase. So by the activity of the first function, what will happen? It will transfer this trisaccharide unit from here to this end, to this non-reducing end, and you will get a structure like this. Now what happens? By the action, second activity of the debranching enzyme, this will be removed. This will be removed as glucose. And then you get this structure. Then again on this, glycogen phosphorylase will act and it will sequentially remove glucose as glucose 1-phosphate. Okay. Now, where is glycogenolysis taking place? This is taking place in the liver and the muscle, and the organelle is cytoplasm majorly. 1 to 2% of this take place in the lysosome and in the smooth endoplasmic reticulum. And it is taking place in the early fasting state under the effect of glucagon. Now, energetics, one glucose residue derived from glycogen gives 3 ATPs. Why? Because in the muscle, you have glucose 6-phosphate, okay? So the first step of hexokinase, which was utilizing 1 ATP, that is not taking place anymore. So instead of getting 2 ATPs, you get 3 ATPs, okay? So from the glucose that is derived from glycogen in the muscle, you get 3 ATPs. Remember this, and not 2 ATPs, because the glucose 6-phosphate is entering glycolysis, okay? And not glucose. Here, glucose 6-phosphate is entering glycolysis and not glucose. Therefore, the first step of hexokinase, which was using 1 ATP, that is not involved, and therefore you get 1 extra ATP, that is 3 ATPs. So that was about glycogenolysis and glycogen synthesis. Now let's take the regulation. Regulation is very important for glycogen metabolism. Very important. Regulation in general for any cycle is very important. Now, your liver glycogen, it is responsible for maintaining the blood glucose level, and your muscle glycogen, it is responsible for providing ATP during muscle contraction. So in liver, glycogenolysis occurs in fasting state, which is very obvious. Why? Because it is involved in maintaining the blood glucose level. And glycogen synthesis takes place in the well-fed state when you have excess glucose more than what the body needs. So you will store that excess glucose as glycogen. Now in the muscle, your glycogenolysis will occur during active exercise. So when you're exercising and the ATP and the muscle needs ATP for contraction, that is when your glycogen in the muscle glycogen will be broken down. And your glycogen synthesis will begin as soon as the muscle is at rest again. Now moving on to the regulation of both glycogen synthesis and lysis. There are two types of regulation, hormonal and allosteric. So we'll take each of them one by one. So first, let's take the hormonal regulation of glycogenolysis. So for glycogenolysis, the enzyme hormone is your glucagon. So glucagon has receptors in the liver, and your epinephrine has receptor both in the liver and the muscle. So your ligand, that is either glucagon or epinephrine, they bind to the respective receptors and they activate what? They activate G protein. Now these G proteins, they will activate what? Adenyl cyclase. So when the ligand binds to receptors that are G protein couples, that is GPCR receptors, your G protein is activated, and this activated G protein in turn activates adenyl cyclase. What will adenyl cyclase do? It turns ATP into cyclic AMP. And this cyclic AMP, it activates what? It activates cyclic AMP dependent protein kinase A into cyclic AMP dependent protein kinase A by it will disassemble or dissociate the catalytic and the regulatory units, as you can see here. So in short, what is happening? If you take this in a flowchart diagram, your glucagon or your epinephrine, it will bind to what? It will bind to G protein coupled receptors, that is GPCRs. This will in turn activate what? G proteins. These G proteins, they activate adenylyl cyclase. And this changes ATP into cyclic AMP. So cyclic AMP level will increase. This cyclic AMP, it activates what? Protein kinase. This is activated. Now this protein kinase, this will activate phosphorylase kinase B by phosphorylating it. You can see this glycogen phosphorylase kinase has been activated by phosphorylation. Now this glycogen phosphorylase kinase A, this will activate your glycogen phosphorylase, which is the rate limiting enzyme, the most important regulatory enzyme of glycogen lysis. So you see this is phosphorylated and it becomes activated. Now what will happen to glycogen is degraded. So that was about the hormonal regulation of glycogenolysis. Now hormonal regulation of glycogenesis, it is just the opposite of glycogenolysis. How? Because on phosphorylation, when your glycogen synthase is phosphorylated, unlike glycogen phosphorylase, it becomes inactive. So your glycogen phosphorylase, when it was being phosphorylated, it becomes active. But when your glycogen synthase is being phosphorylated, it becomes inactive. And this is the reciprocal regulation of both glycogen synthesis and lysis. Similar, it's just the similar thing. Okay, you see, glucagon binds to the receptor, adenylyl cyclase being activated, cyclic AMP level increasing, your protein kinase A being activated. Now this activated protein kinase A, it phosphorylates the active glycogen synthase. You can see has been phosphorylated, and when it becomes phosphorylated, it becomes inactive. And when it becomes inactive, what will happen? Your glycogen synthesis is inhibited because your glycogen synthase is inactive. Okay. So this is the exact opposite effect of glucagon on glycogen synthesis. Now, another important thing, you see this diagram here. What is insulin activating protein phosphatase? Your protein phosphatases are enzymes that remove phosphatase. Okay. So if your protein phosphatase is active here, so it will remove phosphate. So what will happen? Your glycogen phosphorylase, for example, okay, your glycogen phosphorylase, insulin activates protein phosphatase. So protein phosphatases are those enzymes which remove phosphate. So if your glycogen phosphorylase is active and your protein phosphatase removes this phosphate from it, what will happen? It will become inactive, okay? And insulin is doing this. Now let's have a look. If protein phosphatase inhibitors are active, okay? So protein phosphatase inhibitors, what will they do? They will inhibit protein phosphatase. So your protein phosphatase are inhibited, that means they will not be able to dephosphorylate the enzyme glycogen phosphorylase. Thus, what will happen? There will be maintenance of phosphorylation, okay? And this protein phosphatase inhibitors, they are being activated by glucagon. So glucagon is regulating your glycogen lysis in two ways. Firstly, it is phosphorylating your glycogen phosphorylase, okay? Secondly, it is also activating what? Protein phosphatase inhibitors. So this is also involved in maintenance of phosphorylation, okay? So this is just the summary of the regulation. Again, I'll just repeat. to make it more clear. See, your glycogen, it binds to its receptor in the liver. Your epinephrine has receptor both in the liver and the muscle. So your glycogen and epinephrine, they bind to the respective receptors. What happens? Your G proteins are activated, which activate adenylyl cyclase, which in turn increases the cyclic AMP levels. And your cyclic AMP, increased cyclic AMP, they activate protein kinase. And protein kinase, what do? They bring about phosphorylation of the regulatory enzymes. So when your glycogen synthase is phosphorylated, it becomes inactive. When your protein phosphatase inhibitor is phosphorylated, it becomes active. And when your phosphorylase kinase is active, it phosphorylates your glycogen phosphorylase and it becomes active. And your glycogenolysis is increased. So it is very easy, very systematic. You just have to understand the concept. Okay, when you get that right, it will not be confusing. Okay, so just hearing it, and if you haven't understood it, it'll be very confusing. What is getting activated, what is getting deactivated? But if you pause, slow it down, and listen to what I'm saying carefully, and if you get the concept once, this will be very easy. Okay, again, a summary. I've put in pictures, multiple pictures of summary, so that you're able to understand and you don't have to mug it up, because if you try to mug this up, this will be really confusing. Okay, and you're bound to get it wrong. Okay, again, and multiple different flow charts, so that if you're not able to understand one of the diagrams, you might get it in the another diagram. Let's see. Your glycogen synthesis, the important regulatory enzyme is glycogen synthase. Your glycogen lysis, the regulatory enzyme is glycogen phosphorylase. Okay. Now I said your glycogen synthase is under the effect of insulin. Okay. So under the effect of insulin means it will be active in dephosphorylated state. When it is dephosphorylated, it will be active. Now you can see your insulin, it activates protein phosphatases. So for protein phosphatases, they remove phosphate. See, phosphate has been removed. So what has happened? Dephosphorylation has taken place. So your glycogen synthase has been dephosphorylated. Now when it is dephosphorylated, you can see it is activated. Very simple. Now again, glucagon and epinephrine, what do? They bring about phosphorylation. They activate protein kinase, which bring about phosphorylation. You can see phosphate is being added. So what is happening? Phosphorylation taking place. And when phosphorylation, it is glycogen synthase is phosphorylated, it becomes inhibited. You can see it is inactive. Here, it is inactive. Okay. Similarly, you can understand the concept of glycogen phosphorylase now. So insulin, what will it do? It will dephosphorylate by, and how does insulin dephosphorylate? By activating your protein phosphatase. So insulin activates protein phosphatase, which dephosphorylates the enzyme, and your glycogen phosphorylase, when it is dephosphorylated, it becomes inhibited. You can see inactive. Now this is a very simple diagram. I think you're able to understand now, if you haven't been able to understand up till now. Now again, your glucagon, epinephrine, they activate phosphorylase kinase. Phosphorylase kinase, what does it do? It phosphorylates enzyme. So when your glycogen phosphorylase is phosphorylated, you can see this, it becomes active and your glycogen lysis will take place. So that was about the regulation. Hormonal regulation. Okay, now moving on to the allosteric regulation. Now your glycogen synthase, it is activated by glucose 6 phosphatase. So your glucose 6 phosphatase will activate glycogen synthase and your glucose will be converted into glycogen. On the other hand, your glucose 6 phosphate, ATP and glucose, they all inhibit glycogen phosphorylase, okay, and activate glycogen synthase. Now in the muscle, your glucose 6 phosphate is again activating glycogen synthase. And your glucose 6-phosphate, ATP and ATP, they are inhibiting glycogen phosphorylase, and AMP, it is activating glycogen phosphorylase. Okay, so your low energy signals, when the energy is low, substrate availability is low, then what will happen? Your glycogen phosphorylase will take place. Okay, because energy is low, the body needs more energy. So what will happen? Your glycogenolysis should be activated. So your low energy signals, they will activate your glycogen phosphorylase, and your high energy signals, they will activate glycogen synthase. Now, apart from that, there is two other activations as well. That is activation of glycogenolysis by calcium. Okay. So the calcium that is there in the cell, that coming from the endoplasmic reticulum, what happens? It binds to colmadulin or calcium binding protein, and the complex that is formed, that is your colmadulin calcium complex here. This is able to activate number of enzymes, and one of them is your glycogen phosphorylase. So your glycogen phosphorylase is being activated by this calcium colmadulin complex. Now, other way how it can be activated is by activation of glycogenolysis in the muscle by AMP. So your muscle glycogen phosphorylase, it has a binding site for AMP. So when AMP binds to this muscle glycogen phosphorylase, this muscle glycogen phosphorylase, it gets activated without phosphorylation, and this is taking place in case of extreme anoxia. So that was about regulation of your glycogen metabolism. Now, glycogen storage diseases, the clinical associated with glycogen metabolism, it is very important. Your glycogen storage diseases are very important, okay? Especially your von Gerke, okay? This is like the most asked and most important, so you just cannot skip this. Now, what are your glycogen storage diseases? They are a group of genetic diseases due to defect in enzyme required for glycogen synthesis or degradation. So any of the enzyme defective in glycogen synthesis or degradation, it will result in a group of genetic diseases, that is your glycogen storage disorders. Very important from the examination point of view. They result in the formation of glycogen that has abnormal structure, or in excessive accumulation of normal amounts of glycogen in specific tissues as a result of impaired degradation. Okay. Now, the most important glycogen storage disease is your von Gerke. What is defective here? Your glucose 6-phosphatase is defective. So your glucose 6-phosphatase is defective, it leads to glycogen accumulation in liver and renal tubules. So this will cause accumulation of glycogen in liver and renal tubules, will cause what? Hepatomegaly, and in renal tubules it will cause renomegaly. Other important biochemical hallmarks are your hypoglycemia, lactic acidemia, ketosis, hyperlipidemia. So this is type 1A von Gerke. There is another type 1B von Gerke here. The endoplasmic reticulum glucose 6-phosphate transporter is defective. Now, if you remember, when we studied glycogen metabolism, we had studied that your muscles, they lack glucose 6-phosphate. Okay. Only in liver, your glucose 6-phosphatase was present. Okay. But in the liver also, where it was present in the endoplasmic reticulum. So your glucose 6-phosphate, it was entering via a transporter into endoplasmic reticulum, and here it was acted upon by glucose 6 phosphatase and changed into glucose, and via glucose transporter 7 this was coming out. So glucose was coming out. Now in 1 Gurkey type 1b, what is happening? This transporter is defective. Okay, so here glucose 6 phosphatase is not defective, but the transporter is defective. Okay, so again it will lead to what? 1 Gurkey only. Clinical manifestations being hypoglycemia. Why will there be hypoglycemia? Because your glucose 6 phosphatase will not be able to change into glucose. So your glucose level will decrease, leading to hypoglycemia. While there will be lactic acidosis. Why? Because this glucose 6 phosphate now will enter what? Anaerobic glycolysis. So it will form what? It will form lactate, leading to what? Lactic acidosis. Now there will be ketosis as well. Why will there be ketosis? Because your glucose 6 phosphate, it has two fates of either entering anaerobic glycolysis and form lactate, which will form lactic acidosis, and it can also enter into aerobic glycolysis. Now it enters aerobic glycolysis, it will form what? Pyruvate, which changes into acetyl-CoA. Now this acetyl-CoA, it has two fates. It can either enter TCA or ketone body synthesis, but it will not enter TCA because oxaloacetate is depleted. And why is it depleted? Because of excess gluconeogenesis in the previous state, because you're in the early fasting, your glycogen was not available to provide the glucose. So what happened? You entered the second stage of fasting, and there was excessive gluconeogenesis. Because of excessive gluconeogenesis, your oxaloacetate is already depleted. So this acetyl-CoA that is being formed, it is not entering TCA. So what is the other option for this acetyl-CoA? It is to enter ketone body synthesis. So there will be increased ketone body synthesis causing what? Ketosis. Okay. And the other, it is saying hyperlipemia. And there is one more feature as well, uricemia also. Okay. Why? Because your glucose 6 phosphate, it can also enter HMP shunt. So when it enters HMP shunt, you will have more pentosis. So you will have more purines. And more purines means more purine degradation. So you will have more uric acid formation leading to uricemia. And your acetyl-CoA, it can also enter into fatty acid synthesis, leading to what is shown here, hyperlipemia. Okay, so these are the important biochemical hallmarks for von Gurkey's, and this von Gurkey is important. Okay, so you should know. Then apart from that, you have type 2, which is Pompe's disease. What is happening here? This is also a lysosomal storage disorder. Your lysosomal alpha-1 4 alpha-1 6 glucosidase, that is defective here. This accumulation of glycogen in lysosomes. Then you have Corby's, which is also known as Cory's disease. Here your liver and muscle deep branching enzyme. Okay, so your deep branching enzyme is defective here. That is your Corby's or Cory's. And in your limit dextrinosis, your liver, only your liver debranching enzyme, which is your type 3B. In type 3B, limit dextrinosis, only your liver debranching enzyme is defective. And in type 3A, limit dextrinosis, both your liver and the muscle debranching enzyme is defective. Then you have Anderson's disease, where the branching enzyme is defective. And remember, I had given you a mnemonic A, B, C, D. So I told you Anderson, A for Anderson, and what is defective? Your branching enzyme. And C for Cori, and what is defective? D branching enzyme. So A, B, C, D. Anderson branching, Cori D branching. Okay, so this is how you can remember this. Then you have McArdle syndrome. Here your muscle phosphorylase is defective. And Hirsch disease, liver phosphorylase is defective. And Taury disease, your muscle and erythrocyte phosphofructose kinase is defective. Although this phosphofructose kinase 1 is an enzyme for glycolysis, but it comes under glycogen storage disorder. Why? Because your muscles do not have glucose 6 phosphatase. So the glucose 6 phosphate that is formed in the muscle, it is entering what? It is entering glycolysis. So therefore, it falls under Taury's disease. Okay, and it is associated with poor exercise intolerance because your muscles will not be able to provide the ATP needed for muscle contraction. So that was briefly about your glycogen storage disorders. So for today's class, I think we will have to end it here. We've covered most of the important cycles. That is, we've done glycolysis, we've done gluconeogenesis, then we've done glycogen synthesis, we've done glycogen lysis. So we've covered the important 4 cycles in today's class, and I think in the second class we'll be able to finish these cycles off because your Repoport and Lieberberg and Cody's, they're like too small. They'll just take 5 minutes each. Neofructose, galactose metabolism, not much asked, but we'll touch about that also because that is the purpose of the revision class. We'll almost be covering the entire syllabus, even if it is just touching the periphery, because so that nothing in the exams appear to you as you've never heard about it before. And then your TC and HMP, these are also important. So we'll begin by taking TC and HMP in the next class. Okay, so any queries you can ask, and by the end, after we're done with all the cycles, we'll take the MCQs. So for all the cycles, the bare minimum, you need to know the pathways, the sites, energetics, regulation, and clinical being very important, okay? Okay, thanks for watching.